Welcome, dear students. In this session, we discuss 2020 NEET examination botany questions. We start from the first question. Flippers of penguins and dolphins are examples of flippers of penguin and dolphin are analogous organs, that is, organs having different structural organization and they perform same function. Such organs are called analogous organs. Analogous organs indicate what? Analogous organs indicate convergent evolution. Analogous organs indicate convergent evolution. Flippers of penguin and dolphin are analogous organs, that is, organs having same different basic structure but perform the same function. Natural selection is a process in which organisms having favorable variations are selected by the nature. Adaptive radiation, that is, evolution of organisms from a common ancestral stock, and during the evolution they migrate into the different regions of the same geography, that is divergent evolution. They show homologous organs. Industrial melanism, it is of peppered moth. Next question. Some dividing cells exit the cell cycle and enter vegetative inactive phase. This is called quiescent stage G0. This process occurs at the end of G1 phase. That is, the cells which prefer to enter into G0 phase, they quit cell cycle at the end of G1 and enter into a quiescent stage known as G0 phase. In this phase, cells are metabolically active but divisionally inactive. Those cells which do not prefer to divide, they enter into G0 stage or quiescent stage, and the cell enter at the end, the G0 stage at the end of G1 phase. This is a match the following. Inhibitor of catalytic activity. Inhibitor of catalytic activity can be matched with the malonate. It is a competitive inhibitor of the enzyme succinate dehydrogenase. Succinate dehydrogenase is an enzyme taking part in Krebs cycle. The inhibitor of succinate dehydrogenase is malonate. It is a competitive inhibitor. Possess peptide bonds. Peptide bonds are present in proteins. Among the given, collagen is a protein. It is a muscle protein. Cell wall material in fungi, it can be matched with a chitin. Chitin is a polysaccharide. The monomer is N-acetyl glucosamine. It is found in the cell wall of chitin and exoskeleton of arthropods. Secondary metabolite. Resin is a secondary metabolite. So the answer is option 2. Answer is option 2. Floridian starch has structure similar to. Answer option 3. Amylopectin and glycogen. Floridian starch is the reserve food material of Rhodophyceae. Reserve food material of Rhodophyceae, and this Floridian starch show similarity with amylopectin. Amylopectin is a component of starch, and glycogen, the storage material of animal cells. So answer is option 3. If the distance between two consecutive base pair is 0.34 nanometer and the total number of base pair of a DNA double helix in a typical mammalian cell is 6.6 into 10 to the power of 9 base pairs, then the length of DNA is, the length of DNA can be calculated like this: 6.6 into 10 to the power of 9 is the total number of base pairs, and the distance between two base pairs is 0.34 nanometer, that is 10 to the power of minus 9 meter. So to convert into this, it is to convert it into that is 10 to the meter, divide it by 10 to the power of that is 3.4, 0.34 into 10 to the power of minus 9, that is equal to 6.6 into 10 to the power of 9 into 0.34 divided by 10 to the power of 9. Being this 10 to the power of 9 cancelled, 6.6 into 0.34, it is about 2.2 meters, about 2.2 meters. Answer is option 4, 2.2 meter. Next, the process responsible for facilitating loss of water in liquid form from the tip of grass blades at night. From these 3 points, that is loss of water in the form of liquid, and it is from the tip of grass blades, and it occurs at night and in early morning, that is the process is identified as guttation. The process is identified as guttation. What is the cause of guttation? The cause of guttation is root pressure. The root cause of guttation is root pressure. From the wording of the question, that is loss of water in the form of liquid. It occurs during night and at early morning, and it occurs from the grass leaf blade The process is identified as guttation. The cause of guttation is root pressure. Identify the wrong statement with reference to the gene I that controls ABO blood group. So the first statement, the allele I does not produce any sugar, correct? The gene I has three allele, correct? IA, IB, and I. A person will have only two of the three alleles, correct? But the fourth statement is wrong. So the answer is the gene. And if you want a wrong statement, when IA and IB are present together, they express the same type of sugar. They express different types of sugar. IA produce its sugar, IB produce its sugar. That means IA and IB express simultaneously. This phenomenon is known as codominance. That is, in heterozygous condition, if both alleles express their character, the phenomenon is known as codominance. IA, IB, they are heterozygous. Heterozygous condition. In this condition, both of them produces their own character. So it is codominance. So not same sugar, different. Next question. Dissolution of synaptonymal complex appears during diplotin. Diplotin is a substage of prophase 1. During zygotin, the synaptonymal complex appear, and during diplotin, the synaptonymal complex disappear. Synaptonymal complex is a proteinaceous structure formed between the paired homologous chromosomes during zygote. Synaptonymal complex is a proteinaceous structure formed in between the paired homologous chromosomes. They help crossing over. This synaptonymal complex disappear after crossing over. Crossing over occurs in pachytene. After pachytene, the substage is diplotin and the synaptonymal complex disappear. Which of the following is put into anaerobic digester for further sewage treatment? Answer is activated sludge. During secondary treatment of sewage treatment program, it consists of two phases. First phase is aerobic phase. Second phase is anaerobic phase. During aerobic phase, the primary effluent that is obtained from the primary treatment, is subjected to aerobic treatment. During aerobic treatment, that is the secondary treatment, first phase of the secondary treatment. During aerobic treatment, aerobic microbes are allowed to grow in the effluent, for which the effluent is agitated and air is passed through. During the activity of aerobic microbes, because there is plenty of oxygen, aerobic microbes multiply, they grow, and they grow in the form of a flock. or in the form of a mass. Bacteria, fungal mycelium, they're tangled together to form a mass. This mass of bacteria and fungus is called the flock. This flock is allowed to sediment. Sedimented flock is called the activated sludge. This activated sludge is subjected to anaerobic treatment in that is anaerobic sludge digesters. So activated sludge is subjected to anaerobic treatment. Anaerobic treatment is in the anaerobic sludge. Then primary sludge is the sedimented portion of the sewage after primary treatment. Sedimented portion of sewage after primary treatment. Primary treatment consists of two stages, filtration and sedimentation. After filtration, the sewage is allowed to sediment. During this process, sand, grit, pebble and such heavier particles are sediment. These sediments of primary treatment is called primary sediment. Primary sediment. Then next question. The transverse section of plant shows the following anatomical features. 4 anatomical features are given. Large number of scattered vascular bundles surrounded by bundle sheath. Large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Phloem parenchyma is absent. From these 4 anatomical features, it can be very well identified that the organ is monocotyledonous stem. All these anatomical characters are that of a monocotyledonous stem. Then next is by which method was a new breed of Hisardale of sheep formed by using Buchanary eves and Merino rams. Buchanary eve is a breed. Merino ram is another breed. So these two different breeds, animals belonging to two different breeds are crossed together. The method of breeding is cross breeding. It is obtained from cross breeding. In this case, Buchanary eve, that is this breed, female animal is selected from the Buchanary eve, and from Merino ram, the male animal is selected. Eve is a breed, ram is, Merino. Buchanary is a breed, Merino is a breed. Female animal is selected from Buchanary breed, male animal is selected from Merino breed. So it is a cross breeding. Montreal Protocol was signed in 1987 for the control of, it was for the control of emission of ozone depleting substances. Emission of ozone depleting substances, mainly chlorofluorocarbons, CFC, mainly chlorofluorocarbons. This protocol was signed on 16th September of 1987. So 16th September is celebrated as Ozone Day. Next question. According to Robert May, the global species diversity is about, according to Robert May, the global species diversity is about 7 million. Then 20 million, it is given in option C, It is an extreme estimation. 20 to 50 million is an extreme estimation, but Robert May estimated it as 27 million. Then next is choose the correct pair from the following. Choose the correct pair from the following. First pair is wrong because exonuclease makes cut not at specific places. The cut made by exonuclease are not specific. It is here and there. So it is not specific. But the DNA ligases, they join the two DNA molecule. So option 2 is the answer. Polymerase break the DNA into fragments. Wrong. Polymerase joins they catalyze the polymerization reaction, polymerization of nucleotides. Nucleases separate the two strands of DNA. Wrong. The enzyme which separate two DNA strand is helicases, not nucleases. So here the answer is option 2. Option 2. Option 2. Then identify the substrate. Identify the substance having glycosidic bond and peptide bond respectively in their structure. Glycosidic bond is found in polysaccharides and peptide bond is found in proteins. So we have to identify respectively a polysaccharide and a protein. Answer is 1. Inulin is a polysaccharide made up of fructose, and insulin is a protein. It is a proteinaceous hormone. It is a protein hormone. So glycosidic bond is found, that is carbon-carbon bonding. It is found in polysaccharides. Peptide bonds are found in proteins between carboxyl group and amino groups of the adjacent amino acid. So the answer is option 1. Inulin, it is a polysaccharide. Insulin, it is a protein. But here chitin is a polysaccharide, but cholesterol is not a protein. Glycerol is not a polysaccharide. Trypsin is a protein. Cellulose is a polysaccharide, but lecithin is not a protein. Lecithin is a lipid. Lecithin is a lipid. We want first one polysaccharide, second one protein. Only this one agree. First one polysaccharide, second one protein. Chitin is a polysaccharide, but cholesterol is not a protein. Glycerol is not a polysaccharide, but trypsin is a protein. Cellulose is a polysaccharide, but lecithin is not a protein. It is a lipid. Next is which of the following regions of the globe exhibit highest species diversity? Highest species diversity in the world on earth is in the Amazon forest of South America. Highest, richest biodiversity is found in the Amazon forest of South America. Which of the following is not an inhibitory substance? Not an inhibitory substance. Then answer is 2. Gibberellic acid is a plant hormone which promote seed germination. Gibberellic acid is a plant hormone which promote seed germination, but para-ascorbic acid, abscisic acid, and phenolic acid, these are growth inhibiting substances. Abscisic acid is a growth inhibiting hormone. Others, para-ascorbic acid, abscisic acid, and phenolic acid are growth inhibiting substances. Number of substrate level phosphorylation. Number of substrate level phosphorylation in 1/3 of citric acid cycle. In citric acid cycle, in 1/3 of citric acid cycle, that is TCA cycle, tricarboxylic acid cycle, or Krebs cycle, one substrate level phosphorylation occur. One substrate level phosphorylation occur. That is when succinyl coenzyme A is converted to succinic acid. Succinyl coenzyme A is converted into succinic acid. One molecule of GTP is produced. One molecule of GTP is produced. This GTP provides ATP. This GTP provides ATP through substrate level phosphorylation. Substrate level phosphorylation. What is substrate level phosphorylation? When a substrate having phosphate group, is converted into a product, the released phosphate group from the substrate is added to ADP. ADP becomes ATP. This process is called substrate level phosphorylation, or direct phosphorylation, or transphosphorylation. Here, when GTP is converted into GDP, the released phosphate molecule, it is get joined with a ADP and ATP is formed. ATP is formed. That is what is called substrate level phosphorylation. Such a phosphorylation, substrate level phosphorylation, occurs in the Krebs cycle once, only once, when succinyl coenzyme A is converted to succinic acid. A molecule of GTP is formed. It is coupled with a substrate level phosphorylation and produces one molecule of ATP. Then the process of growth is maximum during. The process of growth is maximum during log phase. Maximum during growth. It is related to geometric type of growth. In geometric type of growth, the growth curve is a sigmoid curve. In geometric growth, the growth curve is a sigmoid curve. That is an elongated S-shaped curve. Elongated S-shaped curve. So initial stage of growth is slow. It is called the lag phase, and then the growth rate increases rapidly, called the log phase. Then the growth rate slows down, and this is called the steady phase. Steady phase. So this portion of growth, this portion that is a rapid increase in growth, is called the log phase, which is a phase in the geometric geometric type of growth. Next, how many true breeding pea plant varieties did Mendel select as pairs which were similar except in one character with contrasting trait? How many pairs are selected? So he selected Gregor Mendel, for his hybridization experiment, selected 7 characters and 14 true breeding varieties. 7 characters and 14 true breeding varieties. 7 characters and 14 true breeding varieties. That is, next is identify the basic amino acid from the following. Identify the basic amino acid from the following. Here, valine is a neutral amino acid. That is, tyrosine is a neutral amino acid. Glutamic acid is an acidic amino acid. Lysine is a basic amino acid. Answer is lysine. There are only 2 acidic amino acids, glutamic acid and aspartic acid. Very easy to remember. Their name end in acid. Glutamic acid and aspartic acid. Even though there are 20 proteinaceous amino acid, only two amino acids, aspartic acid and glutamic acid, end in acid. So they are the acidic amino acid. There are 3 basic amino acid, lysine, arginine and histidine. All the rest 15 are neutral amino acid. Here we have to identify the basic amino acid. Then next step. Here, match the following with respect to meiosis. Zygote can be matched with 4 because during zygote synapses pairing of homologous chromosome takes place. Packaging can be matched with 3 because in this substage crossing over occurs. Diplotin can be matched with a chiasmata because diplotin can be matched with a chiasmata formed during diplotin. Diakinesis can be matched with a terminalization, that is terminalization of chiasmata. Terminalization of chiasmata means disappearance of chiasmata. So the answer is A4, B3, C2, D1. Answer is 3. Again, match the following. Iron can be matched with 4 because it is required for the biosynthesis of auxin, IAA, indole acetic acid. Iron can be matched with then zinc can be here, not iron. Iron can be matched with the required for chlorophyll biosynthesis, that is 3. That is chlorophyll biosynthesis. Zinc can be matched with the 4, biosynthesis of indole acetic acid. Boron 2, pollen germination. Manganese, that is photolysis of water. Answer is 4. Iron can be matched with the requires for chlorophyll biosynthesis. Iron and magnesium are required for the biosynthesis of chlorophyll. Zinc can be matched with the IAA biosynthesis because oxygen synthesis requires zinc. Boron, pollen germination. Manganese, photolysis of water. Next is snow blindness in Antarctica region is due to inflammation of cornea due to high dose of UV radiation. Inflammation of cornea due to high dose of UV radiation. That is, over Antarctica, ozone depletion is severe. Ozone depletion is severe. So UVB enter into the atmosphere or reaching the earth. Ozone protect the atmosphere from the UV radiation. So ozone layer protect the atmosphere from the entry of UV radiation. It act as a shield. And protect it from the UV radiation. So UV rays enter through the ozone layer when there is thin areas on ozone. We call it as ozone hole, and this is more prevalent over the Antarctica region. So answer is inflammation of cornea. Then next is ray florets of sunflower. Ray florets of sunflower has inferior ovary. Ray florets of sunflower has inferior ovary. It is inferior ovary if other floral buds are above the ovary. The ovary is said to be inferior and the flower is said to be epigynous. If ovary occupies the highest position, other floral buds originate from below the ovary. The ovary is said to be superior, flower is said to be hypogynous. If the ovary is neither above nor below, the ovary is referred as half inferior. The flower is called perigynous. That is, based on the position of ovary, there are 3 types of flowers. Ovary superior, flower hypogynous. Ovary inferior, flower epigynous. Ovary half inferior, flower perigynous. Flower perigynous. Next, that is it is match the following. Then Clostridium butylicum can be matched with 2 because it yield It is used for the commercial production of butyric acid. Trichoderma polysporum gives cyclosporin A. Monascus purpureus. It is statin is obtained from Monascus purpureus. It is a blood cholesterol lowering agent. Aspergillus niger, it is used for the industrial production of citric acid. A2, B1, C4, D3. Answer 2, 1, 4, 3. Option 3. Then the plant part which consists of two generations, one within the other. Plant part which consists of two generations, one within the other. Here the answer is 1. A and B. A and D. A and D. Because pollen grains inside anther. Pollen grains are haploid. Pollen grains are haploid. Anther is diploid. So pollen grains represent gametophytic generation. Anther represent sporophytic generation. We want two plant parts which consist of two generations within the other. Here anther consists of pollen grain. Anther consists of a pollen grain. Anther is diploid. So represent sporophytic generation. Pollen grains are haploid because they are produced after meiosis from spore mother cell, from microspore mother cell. So they are haploid. So pollen grains are haploid. They represent gametophytic generation and the sporophytic generation. Embryo is diploid. Then ovule is haploid. In angiosperms, embryo develop within the embryo sac. Embryo sac is haploid. Ovule is diploid. Embryo sac is the female gametophyte of angiosperm. Embryo sac is the female gametophyte of angiosperm. It is haploid. So embryo sac is the female gametophyte. Ovule is the megasporangium and it is diploid. It is the sporophytic generation. So ovule contains embryo sac. In angiosperm, embryo sac is retained within the ovule. Ovule, a sporophytic generation, contains embryo sac, a gametophytic generation. So answer is germinated pollen grain with two male gamete. It is a haploid one. There is no diploid structure in it. So it represents only gametophytic generation. Seed inside fruit. Seed is diploid. Fruit is diploid. Seed is diploid. Fruit is diploid. That is, both of them are sporophytic generation. That is, both of them represent the same generation. Here we want different organs having different generation. Next, experimental experimental verification of chromosomal theory of inheritance. It was done by Morgan. Chromosomal theory of verification of chromosomal theory of inheritance. It was experimental proof was provided by Thomas Hunt Morgan by conducting experiments in Drosophila. Then chromosomal theory of inheritance. The theory states that the physical basis of inheritance are chromosomes. That is, genes possess chromosomes, and chromosomes are the physical basis of inheritance. This theory was proposed by Sutton and Boveri, but experimental verification was provided by Thomas Hunt Morgan. The products of reaction catalyzed by nitrogenase in root nodules of leguminous plants. What are the products? Nitrogenase is an enzyme which catalyzes nitrogen fixation, and it occurs symbiotically in the root nodules of leguminous plants. Here the question is, what are the products of nitrogen fixation? The product of nitrogen fixation is ammonia and hydrogen. The product of nitrogen fixation is ammonia and hydrogen. That is, two molecules of ammonia, or two molecules of ammonia are formed from one molecule of nitrogen. So the product of nitrogen fixation, is ammonia and hydrogen, or that is, here it is, nitrogen combines with 8 proton, H plus 8 H plus 8 electron plus 16 ATP. It gives two molecules of ammonia plus hydrogen. After utilizing the energy 16 ADP. The products are two molecules of ammonia and hydrogen. So here it is 4 hydrogen atom participate, that is represented by 8 represented. This represent the nitrogen fix, product of nitrogen fixation. Now then identify the incorrect statement. Here the incorrect statement, due to the deposition of tannins, resins, oil, etc., heartwood is dark in color. Correct? Heartwood does not conduct water but gives a mechanical support. Heartwood is the central part of the secondary xylem, or the central part is called the heartwood. It is dark brown in color. It is due to the deposition of tannins, resins, oil, etc. And the heartwood is dark brown in color. It is the central part of the wood. Then sapwood is involved in conduction of water and minerals. Sapwood is the peripheral part, outer part of the secondary xylem. Heartwood is the inner part of the secondary xylem. So sapwood, the function is correct. Then fourth statement, sapwood is the innermost secondary xylem. it is lighter in color. The statement is due to because of this. Sapwood is the outermost secondary xylem. Sapwood is the outermost secondary xylem. Here we want to select an incorrect statement. Answer is option 4 because heartwood occupies the peripheral part of the secondary xylem. Peripheral part of the secondary xylem. Next, it is in light reaction, plastoquinone facilitate the transfer of electron. Plastoquinone facilitate the transfer of electrons from. Answer is 2 from PS2 to cytochrome B6F because PS2 participating in the non-cyclic electron transport. The electron first move to the primary electron acceptor. From primary electron acceptor, it goes to plastoquinone. From there, it go to cytochrome B6F. Cytochrome B6F. So the electrons are transferred from PS2 to cytochrome. PS2 to cytochrome B6 complex. Among the given, that is plastoquinone, it transfer electron from PS2 to cytochrome B6. Among the given option, it is option 2, PS2 to cytochrome B6F complex. Then, next question. From his experiments, Stanley L. Miller, S.L. Miller, produced amino acids by mixing the following. That is Stanley Miller. He showed the chemical evolution, or he provided from his experiment there is a possibility of a chemical evolution and forming organic molecules. What he used in his experiment, answer is option 3, ammonia, hydrogen, methane, hydrogen, ammonia, water vapor, and it is kept at 800 degrees Celsius. That is, in his flask, in his experimental apparatus, he took some components and he mimicked the atmosphere of primitive Earth. What are the components he took? Methane, hydrogen, ammonia, and water vapor. Methane, hydrogen, ammonia, and water vapor are the chief components of the atmosphere of a primitive Earth. Atmosphere of primitive Earth. So answer is 2. From this experiment, he obtained some amino acid. Then next, select the correct match. Thalassemia is not an X-linked character. Thalassemia is an autosomal disorder. Thalassemia is an autosomal disorder, and the defective alleles are present on chromosome 11 and chromosome 16, not on X chromosome. Thalassemia is an autosomal disorder. It is due to recessive alleles present on chromosome 1 and chromosome 16. Chromosome 1 and 16, because there are two types of thalassemia, alpha thalassemia and beta thalassemia. For one type, it is located on one chromosome. Other type, it is located on the other chromosome. Hemophilia is not a Y-linked character. Hemophilia is an X-linked disorder. Hemophilia is an X-linked disorder. Then phenylketonuria is not an autosomal dominant disorder. Phenylketonuria is an autosomal recessive disorder, and the recessive alleles are located on chromosome 12. They are located on chromosome 12. Then sickle cell anemia. This is correct. This is correct. Sickle cell anemia is an autosomal. Sickle cell anemia is an autosomal recessive trait, and these alleles are located on chromosome 11. The alleles are located on chromosome 11. Then next, embryological support for evolution was disapproved by. Our embryological support, one of the strong evidence from embryology is that during the development of the organism, that is during the ontogeny of the organism, it shows its phylogenetic character, that is ancestral character. This evidence this evidence from embryology is popularly known as ontogeny recapitulates phylogeny. Ontogeny recapitulates phylogeny, also called that is recapitulation theory. This is an embryological evidence, but this embryological evidence in support of evolution was disapproved by a scientist called Carl Ernst Bayer. Carl Ernst Bayer. Then, so the body of the ovule is fused within the funicle. Funicle attaches the body of the ovule at a point called hilum. That is, the point of attachment of funicle with the body of the ovule is called hilum. Then, next, which of the following statement about inclusion bodies is incorrect? Inclusion bodies are reserve food materials of bacteria. Inclusion bodies are reserve food materials of bacteria. Regarding this, we want incorrect one. These are present in cytoplasm. Correct. They are not bound by any membrane. Correct. But these are involved in ingestion of food particle is wrong. They lie free in the cytoplasm. Regarding the inclusion bodies, 1, 2 and 4 are correct. So we want an incorrect statement. So the answer is 3. Answer is 3. What are food vacuoles? Food vacuoles are formed by the ingestion of food particle. It is found in protist. Best example is amoeba. Food vacuoles are formed by the engulfing of ingestion of food materials. Food vacuoles are special type of vacuoles found in protists by the ingestion of or engulfing of the food materials. It is found in eukaryote, and also food vacuoles are surrounded by membrane. Food vacuoles are surrounded by membrane. They are special type of vacuole, especially formed in protist. Answer 3. Name the growth regulator. Name the growth regulator. Name the growth regulator which upon spraying on sugarcane crop increase the length of stem, thus increasing the yield of sugarcane. That is gibberellin. Why gibberellin is used in sugarcane cultivation? Gibberellin is a hormone. The characteristic action of gibberellin, physiological role of gibberellin is internode elongation. Internodal elongation. Elongation of internode. When it is applied in sugarcane cultivation or sugarcane field, internode elongate and the height of the stem increases. Height of the stem increases. What is the advantage of height of stem in sugarcane? Because in sugarcane the sugar is stored in the stem. Sugar is stored in the stem. If the stem part is small, it can store more amount of sugar. It can store, so height of the plant can be increased. Height of the plant can be increased by increasing the internodal region, increasing the internodal length of the internode. The characteristic physiological action is elongation of internode. Elongation of internode. Next is the ovary is half inferior. Ovary is half inferior in plum. Ovary is half inferior. The ovary is half inferior in plum. In brinjal, the ovary is superior. Mustard ovary is superior. In sunflower, the ovary is inferior. In sunflower, the ovary is inferior. So in plum, the ovary is half inferior. Answer is plum. Half inferior ovary, that is intermediate between superior and inferior. Intermediate between superior and inferior. Ovary is half inferior and flower is called perigynous. Flower is called perigynous. Next. Which is important site for formation of glycoproteins and glycolipids. Glycoproteins and glycolipids. Glycoproteins and glycolipids are formed from the Golgi bodies. Golgi body is a cell organelle. It is a component of endomembrane system. What are endomembrane system? Single membrane bound organelles whose functions are coordinated. Single membrane bound organelles whose functions are coordinated. components of endomembrane system are endoplasmic reticulum, Golgi bodies, lysosome, and vacuole. So their functions are coordinated. So here the question is, which is the site of glycoprotein and glycolipid? Glycoprotein and glycolipids are formed in Golgi bodies. What is the main function of Golgi body? Main function of Golgi body is packaging of food material. What is meant by packaging of food materials? Chemical packaging of material. Packaging of material. What is meant by packaging of material? That is chemical modification of material. Chemical modification of a material. Chemical modification of material. Yes. Then why? Endoplasmic reticulum. A rough endoplasmic reticulum produces protein, and that proteins are transported to Golgi bodies, and Golgi body chemically modify it, simply called packaging. They chemically modify the protein by adding carbohydrate. By adding carbohydrate. With the addition of carbohydrate, proteins become glycoprotein. Proteins become glycoprotein. Smooth endoplasmic reticulum. Similarly, smooth endoplasmic reticulum, they synthesize lipids, and these lipids are transported to Golgi bodies, and Golgi body chemically modify the lipid, that is packaging is done. How? By adding carbohydrate with the lipid. After adding carbohydrate with lipid, lipid become glycolipid. Lipid become glycolipid. So glycolipid and glycoproteins are chemical modification of lipid and protein respectively, and this chemical modification, what we simply call packaging, is done by Golgi apparatus. So glycoproteins and glycolipids, the main site is a Golgi body. When these proteins and lipids are sent to Golgi body accept it. So cis phase of Golgi body, cis phase, that is forming phase, it is convex in appearance. The cis phase of Golgi body accept or receive these proteins and lipids formed and sent from endoplasmic reticulum in the form of small vesicles. The vesicles contain protein and lipid, and the vesicles are received by cis phase of Golgi apparatus. Forming phase of Golgi apparatus. Forming phase of Golgi apparatus is the cis phase. After chemically modifying it, it is these glycoproteins and glycolipids are released from trans phase of Golgi body. Trans phase of Golgi body. Trans phase of Golgi body. Trans phase of Golgi body, it is also called the maturing phase. It is concave in appearance. It is concave in appearance. These are the two phases of Golgi body. Cis phase, they accept the molecules to be packed or chemically modified. Then after chemical modification, they are released from the trans phase. So that is the next is identify the correct statement with regard to G1 phase. G1 phase. Identify the correct statement with regard to G1 phase. G1 phase is the first substage of interphase. Interphase is divided into three substages: G1, S, G2. G1, S, G2. Then, what are the events of this substage? In G1, the cell is metabolically active, continue to grow. S, replication of DNA. G2, synthesis of proteins in preparation of the M phase. Here, answer is 4. Cell is metabolically active, grows, but does not replicate DNA. It is correct. In G1 phase, there is no DNA replication. DNA replication occurs in the S phase. DNA replication occurs in the S phase. So that is correct. Then nuclear division takes place. It is wrong. Nuclear division takes place. Nuclear division takes place in the M phase. That is karyokinesis. DNA synthesis or replication takes place wrong. It takes place in the S phase, second substage of prophase. Reorganization of cell components takes place is wrong because it occurs in the telophase. So the fourth option is the correct one. Then next is the first phase of translation is the synthesis of protein according to the information or genetic code given in the or present on the mRNA. That is translation. Translation is the synthesis of protein as per the genetic information present in the mRNA. The translation consists of 4 phases. First phase, aminoacylation of tRNA, that is activation of amino acids and helping them to bind with the tRNA. Then initiation, elongation, termination. The answer is option 4. The first phase is aminoacylation of tRNA. During this phase, amino acids are activated and they are attached to the respective tRNA. Then next is initiation, elongation and termination. Next, name the enzyme that facilitate the opening of DNA during transcription. DNA helix, that is a segment during transcription. A segment of DNA helix unwind. The 2 strands of DNA are separated. Then during transcription, it is done by RNA polymerase. During transcription, it is done by RNA polymerase. But during DNA replication, also the DNA strands unwind. The helix of DNA open. During DNA replication, it is done by DNA helicase. It is done by DNA helicase. They unwind the unwinding of DNA helix or opening of DNA helix. It is done by RNA polymerase in transcription, helicase in DNA replication. The roots that originate from the base of the stem are, that is, there are two types of root system, fibrous and taproot system. Then in monocot stem, the roots originate from the base of the stem. These roots are called fibrous roots. Roots originate from the base of the stem. They are called fibrous roots and they are present in the monocot. Lateral roots are branches of root. Primary root, it is the root which develop from the radicle. Then prop root, which are developed from the stem, it is an adventitious root, developed from the branches of stem, and they give support. The root which develop from the base of the stem is a fibrous root. Fibrous root. Oxygenation activity of rubisco enzyme in photorespiration leads to, that is, when RUBP combines with the oxygen and the rubisco it acts as oxygenase. What is the product? The product is one molecule of 3-phosphoglyceric acid and one molecule of phosphoglycolate. Rubisco is a 5-carbon compound. There is no carbon atom in oxygen. 3-PGA is a 3-carbon compound. Phosphoglycolate is a 2-carbon compound. That is, the 5-carbon compound RUBP is converted into a 3-carbon compound and a 2-carbon compound. From the given option, it is one molecule of a 3-carbon compound. One molecule of 4-carbon compound wrong. One molecule of 2-carbon compound correct. 2 molecules of 3-carbon compound wrong. 1 molecule of 6-carbon compound wrong. It forms 1 molecule of 3-carbon compound, 1 molecule of 2-carbon compound. From the given option, it is option 3. Which of the following is most abundant in? Which of the following is most abundant in animals? Answer is collagen. Answer is collagen. Collagen is a protein. It is a muscle protein. It is a muscle protein. Collagen, it is a muscle protein. Then, so most abundant in plant is rubisco. Most abundant in plant and also most abundant in biosphere is rubisco. Most abundant in plant as well as most abundant in biosphere is rubisco, the enzyme which take part in the dark reaction of photosynthesis. Next, which of the following pairs of unicellular algae? Then unicellular, that is the unicellular one are, that is chlorella and spirulina. Chlorella is a green algae. Spirulina is a blue-green algae. Both of them are unicellular. Then laminaria and sargassum. Both of them are multicellular. Gracilaria and gelidium. Both of them are multicellular. Volvox is multicellular. Amoeba and Anabina, Anabina is not it is a blue-green algae, but not unicellular. It is a filamentous blue-green algae. It is a filamentous blue-green algae. Filamentous blue-green algae. Next, in water hyacinth and water lily, pollination takes place. In water hyacinth Icornia, and water lily, both of them are aquatic plants, but in these plants, pollination occurs by insect or wind. Pollination occurs by insect or wind. Even though they are aquatic plants, pollination is not by water. Water pollination or hydrophily, pollination due to water is a rare type of pollination. It is restricted to only 30 genera of angiosperms. And even some plants, especially water hyacinth and water lily, even though they are aquatic, pollination is by insect or bird. Then next is then which of the following statement is correct? First statement, adenine does not pair with thymine. It is wrong. We want a correct statement. Adenine pairs with thymine through 2 hydrogen bonds. It is correct. Adenine pairs with thymine, 2 hydrogen bonds. Adenine pairs with thymine through 1 hydrogen bond is wrong. Adenine pairs with thymine through 3 hydrogen bond is wrong. Adenine pairs with thymine with a 2 hydrogen bond. Adenine is a purine, thymine is a pyrimidine. Then the nitrogen bases of DNA, adenine, thymine, guanine, and cytosine. Adenine, thymine, guanine, and cytosine. Here adenine, guanine are purines, cytosine, adenine, guanine are purines, and cytosine and thymine are pyrimidines. So each base pair of DNA consists of a purine and a pyrimidine. One purine, one pyrimidine. If the purine is adenine, the pyrimidine will be thymine. Pyrimidine will be thymine. So the pairing is between AT, adenine pairs with a thymine. Then if the purine is guanine, the pyrimidine is cytosine. So cytosine pairs, guanine pairs with a cytosine. Their pairing is with when guanine pairs with a cytosine with 3 hydrogen bonds. Guanine pairs with cytosine with 3 hydrogen bonds. Adenine pairs with thymine with 2 hydrogen bonds. This is the last question of 2000, last botany question of 2020 NEET question paper. So the discussion ends. Thank you.